Okay, this is a podcast on Punnett squares. Our question here is, how can we predict which traits will be inherited by an offspring? So let's say you have a child. What is a probability that the child is going to be tall or short, or is going to have brown eyes or green or hazel eyes? And the answer to that question is that we can predict the possibilities, also known as probabilities, using a tool called the Punnett square. Okay, so here I'm going to go over how to do this step by step. The first step is to read through the problem and underline the key science words. Okay, so the problem says, let's say that Mr. Parks is heterozygous tall. Okay, so I'm going to underline Mr. Parks is heterozygous tall, and Mrs. Parks is homozygous tall. If they have a child, what is the probability that their child will be tall? So usually when you look at problems, it could be overwhelming, but what you do is you want to first convert the problem into its genotypes. So the letters. Mr. Parks is heterozygous tall. So it's telling us that here in this statement, it's telling us that tall is a dominant trait. Okay, because the heterozygous will have a big T and a little t, and the one that is being expressed is the tall. So Mr. Parks is going to be capital T, little t. Let's call T tall. Okay, we're going to say T represents tall. Okay, and little t is going to represent short. Okay, and sometimes in the problem it doesn't tell us, so we might have to make assumptions, and that's what makes these problems difficult. So Mr. Parks is heterozygous. It's easy because heterozygous is capital T, little t, right? Mrs. Parks is homozygous tall. So that means it's going to be capital T, or little t. And because it's tall, it's going to be two capital T's. Okay? So the first step is to underline, to try to figure out what the genotypes are. Also, what is the problem asking for? But usually we tend to worry about that after the Punnett square is drawn. So the third step is to put these genotypes into a Punnett square. So a Punnett square typically is a 4 square, but because we're using a computer here, I have to include these here. So what you do is you put one of the parents on top like this, and the other one on the left side. It doesn't matter which side you choose first, and then you start to move one allele down. So this capital T goes here, and this one goes there. So you're starting to cross them. This is what we mean by cross. Okay, capital T, little t. Always put the capital first. Capital T, little t. So you're crossing them like that. Okay, you're crossing them like a multiplication table. Now, that is step 4. You're bringing the individual alleles from the top and left, and you're combining them like a multiplication table. Step 5 is you're going to count. So for example, I'm gonna put it down here. I'm gonna choose red as my color. So look, here is a 4 square. So I see two that are capital T. So 2 of the 4, which is equal to 50%, is gonna have this combination, this genotype. And we know that capital T is gonna express tall. Okay, then we see that there's 2 out of 4, which is also 50%, that is going to again represent tall, because the capital T blocks the expression of the little t, okay? And so now we want to look at the question. It says, what is the probability that their child will be tall? So I see that 2 out of 4 is tall, and 2 out of 4 is tall again. So that means 4 out of 4 is tall, right? So that's your answer. 100% of the kids are tall. What if it asks, what is the probability it's going to be short? The probability the kid will be short. Then in that case, it would be 0%, right? 0 out of 4, which is equal to 0%, would be short. But the question is asking for tall. So this would be our answer right there, okay? So this is how we do Punnett squares. I know I already included the answer there. I go over another example here, in example 1. Basically, you're doing it again, but for different scenarios. So, you know, what if you have these parents? You're going to put them on the left and on top, and you're going to cross them, and then you're going to count. You know, 2 of the 4 is capital T, 2 of the 4 is heterozygous, and 100% will be tall, okay? I want to do one more just to make sure that you're getting it, which is number 5. This one is saying that round is dominant. So big R we're going to call dominant, and we're going to say it's round. Little r is going to be wrinkled. So here we're talking about P's, and here the genotypes are already given to us. So capital R, capital R, little r. Okay, I just moved these here and these there. Then you're going to cross them, right? Just the way I taught you. Right, there you go. And then you're going to count. So I see only 1 of the 4 are capital R. So 1 out of 4, which is 25%, is capital R, which is going to represent dominant, which is round. Okay, capital R is round. So round here, I see 2 of the 4, which is 50%, are capital R, little r. And again, the dominant one wins, and that's round. So 50% will also be round. And then I see that 1 out of the 4 is little r, and little r is wrinkled. Okay, so the question here is, actually there is no question here. It's just asking for summarizing the results, right? So if we have, if 25% is round here and 50% is round here, that means 75% in general will be round and 25% will be wrinkled. Okay, so that's the way we do these Punnett square problems. If you find another way to do it and it's easier that way, go ahead and do it. But as long as you're answering the question, then you're good, okay? And you're using Punnett squares as evidence, okay? So that's how we predict what probability, what is the chance that a P will be round, what is the chance that a P will be wrinkled, or what is the chance that, you know, the child will be tall, we use Punnett squares to make those predictions. So you're going to be completing this and check your answers, and then you're gonna attempt this multiple times, a second attempt, a third attempt, and then a fourth attempt. Okay, let me know if you need help.